Hi, I'm Gulnar, and today we'll be covering Module 5 titled Communication, Homeostasis and Energy of the OCR A-Level Biology New Specification. Specifically, I will be looking at topic 5.3, which is Neuronal Communication. The objectives of today's video tutorial are as follows. First, I will consider this topic in context of the whole module to encourage integrative thinking, which is certainly a requirement of the new examination papers. Secondly, I will introduce the idea of a nerve impulse or an action potential. Thirdly, how resting potential is established and maintained, and how an action potential is generated and how it propagates. Lastly, I will discuss how stimulus intensity can be decoded. And to finish off, we will look at some exam-style questions to get an idea of what to expect from the new examination papers. And here is a summary of the different components. In general, component 1 is testing for your understanding and for your knowledge through section A with multiple choice questions, of which I will give an example. And in section B, they are looking for you to apply your knowledge a bit more through short answer questions and extended response questions, which are the ones to watch for the extra top marks. And the last exam which this topic which I'll be covering can be tested in is component 3. This is the unified biology component, and here they are looking for how you analyse evaluate and discuss scientific evidence that you are given, and it requires you to think a bit more on your feet. So using what you know, and given this new information in the exam, how you construct an answer or evaluate the different approaches. Because the new papers require such an integrative approach, I will highlight in green different modules or different topic areas which relate to this specific topic, and in pink I will highlight some of the terminology which may be required to gain those easy marks. So now we're considering how neuronal communication fits in with the rest of the module. Communication is absolutely key for monitoring a change in an internal and external environment, responding to this change and coordinating the activities of different organs. This communication can be through either a neuronal system or a hormonal system, i.e. through electrical signals that pass through neurons or chemical signals that pass through the blood. Both systems are vital for homeostasis, which has been covered in specification point 5.1.1. And for example, neuronal signals from thermoreceptors and the hypothalamus are involved in a negative feedback loop, another good terminology there, which is required for maintaining optimum body temperature. On the other hand, control of blood glucose is an example of a hormonal system in homeostasis, and this is covered in 5.1.4. So now we will turn our attention to the actual mechanisms by which impulses are transmitted along neurons. I like to think that a neural signal is similar to a signal you would get when you press a key on your keyboard. But this is because no matter how hard you press it, you'll still get the same size number on your screen, and this number will appear on the screen with the same speed each time. This is kind of similar to an action potential, because the amplitude or the speed of propagation does not depend at all on the stimulus intensity. It is described as an all or nothing electrical impulse. But what is an electrical impulse? The simplest way I thought to describe it is as follows. So the lipid bilayer functions as a barrier which keeps charges separate. This allows the build-up of potential. In a biological system, this is measured in millivolts and is referred to as the membrane potential. So any change in the properties of the bilayer or the barrier will cause ions to move through it, and this will cause changes in membrane potential. For ions to actually move when there are changes in the membrane properties, at rest there must be an asymmetric distribution of ions across the membrane. And this is shown in this figure. As we can see, there is a greater amount of sodium or greater concentration of sodium ions outside the axon or outside the cell than inside, and the opposite is true for potassium ions. Overall, there is a more negative charge on the inside of the cell, roughly around -70 millivolts, but this alters from cell to cell. This inside negative charge is brought about by the presence of negatively charged proteins, and also the fact that there are more sodium ions outside than there are potassium ions inside. One key protein in maintaining this distribution is the sodium-potassium ATPase, which is an active transporter or a pump. This protein transports 3 sodium ions out of the cell and 2 potassium ions into the cell, and a good way to remember this is to think that sodium ion, or Na+, has 3 components to it, and so does out, OUT, whilst potassium ion has 2 components, and so does NIN. As well as this active transport, which is covered in more detail in the biological membranes topic, there's also passive transport of potassium ions because there are open potassium ion channels in the membrane. However, there is no net movement of potassium ions because the movement down an electrical gradient balances the movement out down a concentration gradient. On the other hand, at rest, most sodium channels are closed, so there is very little movement of sodium ions. Okay, so we've discussed how the resting membrane potential is established and maintained at rest, and now we will discuss the importance of it. So if a sodium channel opens, because of the electrochemical gradient, sodium ions will flow in. This will mean that the inside of the membrane becomes slightly more positive. The important value of the membrane potential that we have to remember is -55 millivolts. This determines whether there is an action potential or not. So the reason that this value is so important is because it is the potential at which voltage-gated sodium channels open. So if you get a depolarization less than this value, i.e. More negative, then you get a graded potential. It's called this because, unlike the action potential, which is all or nothing, the graded potential displays a graded depolarization in response to stimulus intensity, but it doesn't trigger the sending of a signal all the way down the axon. On the other hand, when this threshold is passed and the depolarization is large enough to open voltage-gated sodium channels, a positive feedback mechanism kicks in. This serves 2 purposes. Firstly, it means that the action potential is of the same amplitude each time because eventually all the voltage-gated sodium channels are opened by sodium coming in, causing depolarization and causing more voltage-gated sodium channels to open. And also it enables the propagation of this action potential down the axon without decaying, because the entering of sodium ions causes the opening of adjacent voltage-gated sodium channels, meaning there is a self-perpetuating signal that travels all the way down the axon. Okay, so now we'll have a look at the graph at the bottom of the whiteboard, which shows the membrane potential changes during a graded potential or an action potential. So as you can see, the graded potential doesn't reach the threshold of -55 millivolts. However, the action potential does, and this is followed by a rapid rise in membrane potential, which is referred to as a depolarization, and this results from the influx of sodium ions. Following this, which reaches around +40 millivolts, there is a repolarization phase due to the opening of voltage-gated potassium channels. And if we recall from the middle figure on the whiteboard, the opening of potassium channels will lead to the efflux or movement of potassium ions outside the cell because of an outward concentration gradient. And since the membrane potential inside the cell is now positive, this movement will be reinforced by an electrical gradient. This repolarization goes slightly too far and results in a hyperpolarization, which is a more negative membrane potential than the resting membrane potential, and this serves as a refractory period, which ensures that the impulses are only passed in one direction, because a hyperpolarization makes it all the more harder to reach threshold. Following on from this, a good question to think about is, why do different cell types have different resting membrane potentials? So my answer would be that a more depolarized resting potential will mean that that cell is more sensitive to triggering action potentials. So this is desirable in the heart pacemaking cells, the sinoatrial cells, because you want an action potential to be triggered quite easily, because the rhythmic action potential generation underlies pacemaking. Whereas in ventricular myocytes, which are the contractile units of a cardiac cell, you have a much more negative resting membrane potential, because you want them to only contract when an adequate stimulus has been received from the sinoatrial node. This is covered in more detail in the transport in animals section of the syllabus. Going back to the action potential, there's one last thing to note, and that is the sodium-potassium pump activity returns the hyperpolarized membrane potential back to resting. The last thing to discuss is, how do we actually obtain information about the stimulus intensity when action potentials are always the same? I.e., they have the same magnitude and speed because they are an all or nothing response. The answer is frequency. So a low strength stimulus will trigger a low frequency of action potentials, whereas a high strength stimulus will trigger high frequency of action potentials. Okay, so now it's time to answer some exam style questions. So which of the following describes the process that happens during repolarization of a neuron during the action potential? If you remember, the opening of voltage-gated sodium channels actually causes depolarization and initiates the action potential, but for the repolarization phase, these channels close again, and it is the potassium channels which open, allowing potassium efflux to bring the membrane potential back down towards the negative side of the graph. In this case, we would consider the membrane potential to be decreasing. So now let's look at an extended response question that you'd probably find in section B of component 1. This question says, describe and explain how the resting potential is established and how it is maintained in a sensory neuron. So because it says describe, I would first state that the resting membrane potential is the potential difference across the membrane when the neuron is at rest, roughly around minus 70 millivolts, and it is due to the asymmetric distribution of ions either side of the membrane. And now we should go on to explain how it is maintained, and this is through both active and passive transport. The sodium-potassium pump actively pumps 3 sodium ions out and 2 potassium ions in. And then, because the potassium channels are open, potassium can passively diffuse out down a concentration gradient or in down an electrical gradient, but there is no net movement. On the other hand, the membrane is much less permeable to sodium ions, and this is very important because it enables this potential to be harnessed when voltage-gated sodium channels are open during the action potential. It might also be worth mentioning that there are a large number of negatively charged proteins inside the cell which contribute to making the inside negative membrane potential. 3 of the terms in pink must be written down for an extra mark. It is definitely important to realise that they don't give you a mark for just writing the sodium-potassium pump actively pumps sodium. You must say sodium ions, or just write down Na+, which is what I would do to save time and be safe. And then you must include the words as much as possible, active transport or passive diffusion, and lastly, use comparative words where possible. For example, the membrane is less permeable to sodium ion. Thank you for watching this tutorial. Please do excuse the terrible filming and editing, but I hope you enjoyed it anyway.